Hello everyone, welcome back to another video of MRB Pharmacist Tamilnadu Master Stroke series. So dear students, as you all know, for the preparation of this pharmacist vacancy, we are having a Master Stroke series running in which you are getting videos from each and every subject with topic wise and their explanations. So today our topic will be biochemistry and clinical pathology, and the topic of today's this subject will be carbohydrates. So let's get started with some carbohydrate questions. Carbohydrate topics you can also have from the questions. Like first of all, topic, what are the topics we are going to cover? So we will be having carbohydrates. So the first question we are having, the general formula of a monosaccharide is. So for the monosaccharide like glucose, galactose, like for fructose, we are having formula of CN, H2N, and we are having ON. So we are having CN and H2O ka whole N. Okay, so correct. So like for the case of glucose, we are having C6H12O6. So the number of carbons and the number of oxygens are equivalent, and the number of hydrogens are just double. So yeah, the first option is correct. So A will be the right answer. Here A will be the right answer. In case of like glucose, fructose, all like mannose, like galactose, we are all having the same configuration, same atomic configuration. The next question we are having, a pentose sugar is. So a pentose sugar means a sugar which consists of 5 carbon. So 5 carbon rings, like 5 carbon structure, like a dihydroxycetone. It having a 3 carbon system, so it will be a triose. Like a ribulose. Ribulose is a 5 member, so it will be a 5 carbon structure. So it is a 5 carbon structure, so it will be a ribulose. Like erythrose. In erythrose, we are having 4 carbon system. It will be a 4 carbon system, so it will be a tetrose. So triose, tetrose, pentose. And in case of glucose, so glucose, fructose, galactose, mannose, all will be having a 6 carbon ring, 6 carbon structure. So it will be a like hexose sugar. So the right answer for the pentose sugar, it will be ribulose. That is B will be the right answer. B will be the right answer. Okay, for the MRB Pharmacist Tamilnadu, we are also having free of cost test series running. As you all know that we have discussed in earlier videos too. Like you simply have to visit the GDC classes application and enter the batch code like MRB25 in that batches section, and you will be added to the very next test series. You will be you can join from the very next test series. So next question we are having, a heteropolysaccharide among the following is. So heteropolysaccharide, we are having a heteropolysaccharide, like it will be inulin, cellulose, heparin or dextrin. So when we are talking about a heteropolysaccharide, like heteropolysaccharide, it's a polysaccharide structure, means it will be consisting of more than multiple monosaccharide units. It will be a repetitive monosaccharide units, and it's a heteropolysaccharide, means it will be having more than one kind of sugar, more than one kind of polysaccharides. So it will be a, since it consists of more than one kind of sugar, it will be a heteropolysaccharide. And inulin is a homopolysaccharide. Like cellulose, it's also a homopolysaccharide. Like dextrin, so dextrin is also a homopolysaccharide. But in case of heparin, so heparin is a heteropolysaccharide. Heparin is a heteropolysaccharide. So C will be the right answer. Like homopolysaccharide like starch, dextrins, inulins, glycogen and cellulose, they all comes under the category of homopolysaccharide. While in case of heteropolysaccharide, we are having like hyaluronic acid, like a chondroitin, like a heparin. These all comes under the category of heteropolysaccharides. Okay, so they are consisting of more than one kind of like sugar units. Now the next question we are having, alpha D glucose and beta D glucose are. So alpha D glucose and beta D glucose. So alpha and beta are a form of like when the cyclic form of sugar, and the like a Fischer the structure, that is chain structure. The chain structure when converted into the cyclic structure of like glucose or fructose, this is known as mutarotation. This is known as mutarotation, the interconversion between a chain structure into the cyclic structure of that particular sugar. So this is known as, this phenomena is known as actually the mutarotation. Now in this mutarotation, we are having alpha and beta form of a single kind of we are having, let's suppose, glucose. So glucose is actually converted into cyclic form, that is it will be converted in two forms, like one will be the alpha form and one will be the beta form. And this alpha and beta forms of that particular glucose, this comes under the category of anomerism. And these structures, like the same compound converts into two different forms, that is actually known as the anomerism, and they both compounds will be anomers of each other. So they are actually anomers. They are actually anomers of each other. They are anomers of each other. So C will be the right answer here. C will be the right answer here. So like here you can see that we are having alpha D glucose and we are having beta, we are having beta D glucose. What is the major difference between them? So basically the major difference is like the OH group on the very first chiral carbon. So the very first chiral carbon just after the oxygen, like here we are having a oxygen, and very first chiral carbon, it will be having the position of OH different on both the structures. Like here you can see that the OH is actually present on the right side, or you can say that downwards of the down at the bottom side of the ring. So it will be alpha form, and when the OH group is on the left side, or you can say that the upward direction, it will be the beta form of that particular glucose. So they are alpha form and beta form of particular glucose structure, and they only arises when actually the muta-rotation takes place. Now next question, we are having two sugars which differ from one another only in configuration around a single carbon atom are termed as. So the compounds that actually differ in the stereochemistry, or you can say that the configuration at only a single chiral carbon. So what do you name such kind of structures? So these structures are actually known as epimers of each other. They are actually epimers of each other. They will be epimers of each other, like in case of glucose, galactose and mannose. So basically glucose and galactose are C4 epimers of each other, and glucose and mannose are C2 epimers of each other. They will be C2 epimers of each other. So what will be the correct answer? So we will be having, it will be A, that is epimer. A will be here correct answer. A will be the right answer here. A will be the right answer here. Now next question, we are having sucrose consists of. So sucrose will be consisting of sucrose. So like sucrose is a disaccharide. Sucrose actually consists of glucose and glucose and glucose and glucose, glucose and fructose, glucose and galactose, or glucose and mannose. So like when we are having glucose plus galactose, this disaccharide will be known as lactose. It's the lactose sugar. When we are having glucose plus glucose, it will be a mannose sugar. So it's a mannose, glucose plus galactose, it will be lactose. And when we are having glucose plus fructose, this kind of disaccharide will be, this type of disaccharide will be sucrose. So yes, B will be the right answer here. Glucose plus glucose will be constituting like mannose, a disaccharide, that is mannose. Glucose plus galactose will be constituting lactose, that is also known as milk sugar. And when we talk about glucose plus fructose, it will come under the structure like what? Sucrose. Okay. Now next question, we are having which one of the following is a disaccharide? What will be the disaccharide? Which will be the disaccharide present here? Sucrose, fructose, glucose or galactose. So when we are talking about disaccharide, glucose or sucrose. Sucrose is a disaccharide constituting only glucose plus fructose molecules. Like fructose, it's a monosaccharide. Glucose, monosaccharide. Galactose, it's a monosaccharide. So yes, the right answer will be sucrose. Like sucrose will be constituting of glucose plus fructose molecules. Glucose plus fructose molecules. Like disaccharides, we are having the sugars that are hydrolyzed into two molecules of monosaccharide like sucrose, lactose and maltose. So what will you getting? What kind of monosaccharide units are you going to get when we disintegrate, or you can say that we hydrolyze the maltose molecule? Okay, like triacylsaccharides, we are having raffinose and ketoses, and in case of tetrasaccharides, we are having lignose and maltotetrose. Now next question, we are having which of the following are polysaccharides? Which of the following are polysaccharides? It's a polymer, acid, proteins or oils. So when we are talking about polysaccharides, that is more than one sugar units, or you can say carbohydrate units. Which of the following will be polysaccharides? So the right answer is polymers. Right answers will be polymers. So polymers, actually polymer of more than one type of repetitive sugar units, that will become under the category of polysaccharides. That is more than one sugar units when constitute the structure, that will be known as actually the polysaccharides. Okay, so polysaccharides are polymers of monosaccharide units with high molecular weights. Next question, we are having a polysaccharide which is often called animal starch. Which polysaccharide is also actually termed as animal starch? So when we are talking about animal starch, so what do you mean by starch? Starch is the storage carbohydrate in what? Comment down. Like glucose, inulin, starch or glycogen. So when we talk about animal starch, so actually animal store the glucose in form of glycogen. And what actually hormone is responsible for the preparation of glycogen? So this glucose when converts into glycogen, Glycogen. So write down what hormone is going to be participating in these kind of reactions, which type of hormone, what hormone, and this process, or you can say this method or this phenomena is actually known as. This phenomena will be known as. So comment down both the answers. Okay, check yourself. So the right answer will be D, that is glycogen will be the right answer. So glycogen is stored inside the human body, or you can say the animal body. The animal is actually store the glucose in by converting it into the glycogen, and this glycogen is you can say that they are the storage unit for the carbohydrates inside a animal cell. Like maltose is a disaccharide consist of. So maltose will be consisting of like glucose plus fructose. So they are, we have already discussed, it will be sucrose. Like glucose plus galactose, they are lactose. Glucose plus sucrose, no, not at all. That's all. This one is already a disaccharide. Like glucose plus glucose, yes. So the maltose actually constitute glucose plus glucose molecules. When it get hydrolyzed, it will be giving two glucose molecules. All right. So MRV pharmacist study material. So if you are also wish to have the best content for this kind of examination, so you should visit GDC classes application and check for these two books. Like one is a theory and other one is a objective kind of book, where you can have all the syllabus, the detailed syllabus of this examination covered in both the books, with theory as well as the objective type of questions. You can practice as much as you can. Like for the test series also, you can get test series, that is also MRB pharmacist online test series, which constitute 200 plus like premium test and 35 plus mini test with this courses, or you can say you can also opt for these kind of courses inside that application. And we are also having a Tamilnadu MRB pharmacist crash course, where you can join for the complete preparation of this examination. All right, for the next question we have, which of the following is a homopolysaccharide? So homopolysaccharide, the polysaccharide that actually constitute, or you can say that the polysaccharide that is actually made up of only one kind of sugar unit, that is a homopolysaccharide. So heparin, it's a hetero, you can say that it was a hetero. Like inulin, yes, it's a homopolysaccharide. Like chondroitin sulfate, so chondroitin is also a heteropolysaccharide. Like hyaluronic acid, yes, it's also a heteropolysaccharide. So inulin is a homopolysaccharide unit. Okay, inulin is a homopolysaccharide. So yes, it's a right answer. Like starch, dextrin, inulin, glycogen and cellulose, they all are homopolysaccharides. Next question we have, cellulose is made up of repeating units of. So cellulose is actually made up of what kind of repetitive units? So cellulose, it's a, you can say that it's a plant. Cellulose is actually found in plant cell wall. So cellulose is made up of beta 1,4 linkage between D-glucose units. Like alpha 1,4 linkage between D-glucose units, or alpha 1,2 linkage between D-glucose units. So actually it's a D-glucose unit. So D-glucose units, a complete, you can say that repetitive, chain, a repetitive unit of these beta D-glucose at 1,4 attachments. At 1,4 attachments actually constitutes this cellulose. So 1,4 linkage between D-glucose units actually constitute this cellulose structure. Like you can see that here we are having a D-glucose structure. So at D-glucose structure, this will be first position, as I already told you that the first carbon after this oxygen will be the first. So this first is actually joined by the other one's fourth, fourth carbon. So this is a 1,4 linkage. This will be the 1,4 linkage. So what kind of linkage is this? What do we name this kind of linkage? Comment down. What kind of linkage is this? Okay, all right. So let's have the next question we are having. Which of the following is the smallest carbohydrate triose? Which will be the smallest carbohydrate triose? So we are having a triose, means the carbohydrate that is actually made up of 3 carbons. So ribose is a 5 carbon structure. Like glucose, 6 carbon structure. Glyceraldehyde, okay, glyceraldehyde and dihydroxyacetones, like dihydroxyacetone. So like we are having carbohydrate like trioses. So we are having 3 carbon structure. So in case of glyceraldehyde, we are having a 3 carbon structure. In case of glyceraldehyde, we are having a 3 carbon structure, aldehyde with carbon and aldehyde and carbon and methyl group, like methanol groups. So yes, glyceraldehyde is a 3 carbon structure. Glyceraldehyde is a 3 carbon structure. Okay, 3 carbon structure. So why not dihydroxyacetone? Why not dihydroxyacetone is also correct. So dihydroxyacetone is actually having a ketone moiety. It's a ketonic moiety. So yes, the right answer you can go for the more appropriate, that will be a glyceraldehyde. That is actually thought to be the simplest carbohydrates. Okay. All right. The next question we are having: which of the following is an example of a C4 epimer? So C4. So C2 and C4. There are two kind of epimers that you generally have to learn about, the 6 carbon carbohydrates. So we are having like for 6C, like 6 carbon structure for 6 carbon carbohydrates like glucose, mannose and galactose. We are having there will be a C2 epimer. One of them is C2 epimer and the other one will be like a C4 epimer. C4 epimer. When we talk about C2 epimer, it is none other than glucose plus mannose. Glucose plus mannose. So glucose and mannose, actually glucose and mannose are C2 epimers. Glucose and mannose are having C2 epimers. And what do you mean by this epimers? Epimers means that the configuration of these both the carbohydrate units are actually differing at just a single chiral configuration. The configuration, just a one carbon configuration is differentiating them from each other. So yes, the glucose and mannose are C2 epimers, while the glucose and galactose are C4 epimers. C4 epimers. So glucose and mannose, look for the option we are having. Okay, so since okay, for C4 epimer, okay, we are having asked about the C4 epimer, so it will be glucose and galactose. It will be glucose and galactose. It will be glucose and galactose. C4 glucose and galactose. C2 are having glucose and mannose. Glucose and mannose. Okay, look for these structures. So this is a glucose, this one is mannose, this is galactose. Now look. We are having this and this. First compare between the glucose and mannose. So in the case of glucose and mannose structure, look for the only 1 point difference. 1 point difference, like we are having OH on the right side here at the second position, while on this mannose we are having OH on the left side. Okay, rest of the structure like carbon OH. Okay, OH, H, 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 OH, 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 H, H. Okay, then all other are just exact copy of each other. So they are actually identical, but they are actually different at only C2 position. While in the case of glucose and galactose, the whole structures are similar except for the C4th position. So at the C4th position, we are having OH on the left on the galactose, while here we are having OH on the right side. Just a single chiral carbon configuration different make the both structures epimers of each other, and they are also can be termed as they will be diastereoisomers of each other. They are different images. They are different images structures. Okay. Now we are having glucose upon treatment with nitric acid yields. So when we treat glucose with the nitric acid, it will be giving you like a gluconic acid, glucaric acid, glucitol or hepatanoic acid. Okay, so they are not the correct answers, like gluconic acid and glucaric acid. So gluconic acid is actually none other than when aldehyde carbon is actually oxidized to the like acid. So when aldehyde, that is CHO, this carbon oxidized in acid, that gives the structure of gluconic acid. Rest of the structure is exactly same for the glucose, and here we are having gluconic acid. When we talk about glucaric acid, so glucaric acid is actually having like we are having OH, H, OH, OH and CH2OH, H, H, OH, H. This is actually glucose. So this is glucose. Glucose oxidized when this aldehyde group is actually oxidized to the carboxylic, that gives you gluconic acid. And when this CH2OH carbon also oxidized, when this also oxidized in acid, it will be giving you glucaric acid. It will be giving you glucaric acid. So when this just aldehydic carbon is actually oxidizing to the acid, it will be gluconic acid. And when the both terminal carbons actually oxidized to the acids, that is glucaric acid. So basically nitric oxide, that is nitric acid oxidizes both the carbons. So they will be giving you B, that is glucaric acid. They will be actually giving you glucaric acid. Okay, glucaric acid. Like here you can have a screenshot, glucose Br2H2O, it will be only giving you one carbon oxidation, rest the structure will be same, that will be gluconic acid. And when we will be treating this with HNO3, it will be having both the terminal carbons oxidized inside the like carboxylic, so it will be a glucaric acid. All the following are homopolymer polysaccharide, like homopolysaccharide, except all are homopolysaccharide except one, like starch, glycogen, we are having cellulose and heparin, except, so we have already discussed, like heparin is a heteropolysaccharide. So yes, this will not be the right answer here. Heparin is a heteropolysaccharide. D-fructose on simple reduction gives, like D-fructose on simple reduction gives only mannitol, only sorbitol, mixture of mannitol and sorbitol, and L-fructose. So no, this L-fructose is given by none of the compounds, like only mannitol. So this mannitol is actually given by mannose. This will be given by mannose. And when we talk about the sorbitol, sorbitol is obtained from glucose. This is obtained from glucose, while mannitol and sorbitol both can be obtained by the fructose. So fructose gives mannitol as well as sorbitol, mixture of both the compounds via reductions. So yes, C will be the right answer. C will be the right answer. Like glucose gives sorbitol, galactose gives dulcitol, like mannose gives mannitol, fructose gives mannitol and sorbitol both, ribose gives ribitol. They are the reduced product of them. Reduction, on reductions they give these kind of products. Pentose sugar gives which type, which test positive? Like this is a pentose sugar. And pentose sugar gives which test positive? Like inversion test? No, not at all. Mucic acid test, full saturation test, or Bial's test. So this pentose sugar, we are having pentose sugar. So pentose sugar will be giving this Bial's test. So D will be the right answer. Why Bial's test? Pentose react with strong HCl to form furfural. They will be forming furfural derivative, which in turn react with orsinol to form green colored complex. This Bial's test is useful for the detection of xylose in urine, essential for pentosuria. While mucic acid test, so galactose only gives this mucic acid test. Galactose gives this mucic acid test, and galactose is a 6 carbon structure, that is a hexose. Like which of the following is a mucopolysaccharide? So mucopolysaccharides is the other name for the heteropolysaccharides. It's the other name for heteropolysaccharides. So cellulose, D-glucuronic acid, hyaluronic acid, or glycogen. So you can see that yes, this hyaluronic acid is actually a like heteropolysaccharide. So yes, C will be the right answer here. So this heteropolysaccharide is having this hyaluronic acid, and that will be the right answer. Next we are having, what is the major function of carbohydrates? What is the major function for the carbohydrates? Water, circulation, mineral transport. In the mineral transport, storage of energy, or none of the above. So carbohydrates are actually the storage units. By glycolysis and Krebs cycle actually produces energy. So yes, they are actually the storage units. So yes, C will be the right answer. Carbohydrates participate in the wide range of functions, like the major, or you can say the main important function of them is to produce energy. Next question we are having, lactose is made up of. So lactose will be made up of glucose and galactose unit. So one glucose molecule and one galactose molecule. Yes, A will be the right answer. Two glucose molecule, that's mannose. One glucose and one fructose, that's sucrose. Two galactose molecules, no, not at all. So A is the right answer. Lactose will be made up of glucose and galactose. Next question we are having, which sugar gives a positive Selivanov test? Which will be giving you the positive Selivanov test? So Selivanov is actually used for the ketohexoses. So glucose, lactose, or maltose, they will be having like this. Glucose is actually having glucose. Now this lactose is having glucose plus galactose, and the maltose is having only glucose and glucose. So all are having aldehydic groups, aldehydic. while this fructose is having a ketone moiety, ketonic functional group. So yes, B will be the right answer. Like this Selivanov test is going to only positive for the fructose, since only fructose is having ketone group. Only fructose is having ketone group. Okay, so Selivanov test is specifically a specific test for the ketohexoses. Ketohexoses. The percentage of total calorie obtained from carbohydrates should be in between. So the percentage of like you can say that the calorie obtained will be 10 to 20% from the carbohydrate, 50 to 60%, 20 to 30%, or 5 to 10%. So they will be giving you calorie ratio, like they will be having 50 to 60% energy. They will be giving you 50 to 60% of the calorie. Okay, so B will be the right answer. Total calorie comes from the carbohydrate, so they will be having according to US FDA recommendations, like 45 to 65% of your total daily calories come from the carbohydrates. Chemically cane sugar is, so chemically cane sugar is actually made up of like lactose, glucose, sucrose, or starch. So it's none other than sucrose. When we talk about the cane sugar, it will be only made up of sucrose. It will be having sucrose as a unit. So sucrose is made up of glucose plus fructose units. Okay, it will be a disaccharide of glucose plus fructose. Identify the given structure. So what do you name this kind of structure? So we are having 1, 2, 3, 4, 5, 6 carbon, with second carbon is of ketone functional group. This is a ketonic functional group. So since it's a ketone functional group, it's having ketone moiety, so it will be a fructose. It's actually structure of fructose. It is having glucose, so it will be having aldehyde functional group, aldehydes, while this is a fructose that is only having a ketone functional groups. B will be the right answer. Like next question we are having, glycogen is which type of starch? So glycogen is which type of starch? It's animal starch, plant starch, both A and B, or none of the above. So plant starch, no, it's not a plant starch. Glycogen is actually present in animals, so it's an animal starch, you can say. Animal carbohydrates, animal storage units of carbohydrates. Okay, animal starch. Okay, where it is present? It's present in mostly liver and muscles. Which sugar is carried by blood and used by the tissue? So what type of sugar your cells actually used? So maltose, starch, glucose, or hyaluronic acid. So just simple answer, like glucose is the only form of energy that a human cell can consume for its energy, synthesizing. So yes, C will be the right answer. Like glucose will be the right answer here. Glucose will be the right answer here. Okay, so glucose is extracted from the blood and sustain metabolism in variety of tissues. Okay, next question we are having, Felling's test is to identify which type of sugars. So Felling's test, like Felling is actually made up of two kinds of Felling solution. We are having Felling A and Felling B. Felling A constitutes of copper sulfate plus caustic soda, that is NaOH, and Felling B is having Rochelle's salt, that is sodium potassium tartrate. So these both Felling A plus Felling B actually constitutes the Felling solution, and this Felling solution is actually used by the reducing sugars. So they will be used by the reducing sugars. It will be used for the reducing sugar. Reducing sugars are those that are actually having a carbonyl groups. Carbonyl derivatives. They are actually having a carbonyl derivatives. A sugar alcohol is, so a sugar alcohol. So when we talk about the sugar alcohol, so we are basically talking about the sorbitol, mannitol. Okay, so we are having mannitol. Okay, here we are having the right answer. Next we are having trehalose. No, not at all. We are having xylose. No, arabinose. Okay, so we are having mannitol. Right answer. So A will be the right answer here. Like mannitol is a type of carbohydrate called a sugar alcohol, or you can also say that it's a polyol. Now, next question we are having, the major component of liquid glucose is dash and is prepared from. So, for the when we are talking about the major component for liquid glucose, so it will be maltose, dextrine, dextrose, or pectin. So you have seen in various of the hospitals like dextrose. That is, dextrose will be the liquid glucose. Glucose. So yes, C will be the right answer. That is, dextrins, dextrans or dextrose. The first two are polysaccharide composed of glucose. Dextrins are the breakdown products of starch, while dextrans are the gel produced by the bacteria from glucose. Dextrose is a glucose in solution, that is dextrorotatory. So solution, that is a liquid glucose, will be dextrose. Dextrose, that is actually used in various medical practices. So these are the questions from the carbohydrates that we have discussed in this video. I hope you enjoyed them and you have been most answers correctly marked here. So thank you so much for watching this. We will be again meeting you with some new questions inside a new topic. Alright, so thank you all.